The overall goal of this method is to isolate the G-quadruplex helicase G4 Resolvase1 protein from a bacterial expression system using a unique ATP-dependent purification step to isolate nearly pure and catalytically active enzyme.
This method can help researchers obtain nearly pure and catalytically active recombinant G4 Resolvase1 protein for use in a wide range of in vitro biochemical assays. The main advantage of this technique is that it excludes misfolded or otherwise catalytically inactive enzyme from the protein preparation.
The idea to use ATP to elute the G-quadruplex helicase originated after being unable to elute the catalytically active enzyme off the G4 beads, even at exceptionally high salt concentrations.
Prepare and grow large bacterial cultures expressing G-quadruplex Resolvase1 or G4R1 as described in the text protocol. Thaw the bacterial pellet in 3 ml of TN buffer at room temperature. Hold the bottles in hand and swirl to thaw and resuspend the bacterial pellet. Once thawed and relatively evenly suspended, bring the volume up to 5 ml with TN buffer. Next, dissolve 20 mg of lysozyme in 250 microliters of water per culture and add to the resuspended bacteria. Allow the lysozyme to digest the bacteria for approximately 5 to 10 minutes while swirling the bottles in hand. The color of the suspension will begin to lighten slightly as the digestion proceeds, and the suspension will be relatively non-viscous. Add 250 microliters of protease inhibitor cocktail, abbreviated PIC.